Hi everyone, good day to all. In connection to the previous session, today we are going to discuss about measurement of bioavailability. So in the earlier class, we have been discussed about what is bioavailability, what are the various objectives as well as significance of bioavailability, followed by consideration parameters during bioavailability such as single dose versus multiple dose, absolute solubility, absolute bioavailability and relative bioavailability, patient versus healthy volunteers. So these all we have been discussed in the previous class. So today we are going to discuss about measurement of bioavailability. Well, in measurement of bioavailability, the methods useful in quantitative evaluation of bioavailability can be broadly divided into two categories. One is pharmacokinetic method and the other is pharmacodynamic method. So in case of pharmacokinetic methods, these are very widely used and based on the assumption that the pharmacokinetic profile reflects the therapeutic efficacy of a drug. So thus these can be called as an indirect estimation or indirect method. The two major pharmacokinetic methods are plasma level time studies, urinary excretion studies. Well, the next one is about pharmacodynamic method. These methods are complementary to pharmacokinetic approach and it involves direct measurement of drug effects on the physiological process as a function of time. So in various time intervals, what is the drug concentration available in the blood or in the physiological area? So that's what nothing but a pharmacodynamic method. Here saying in other way, what is the response of the drug? What is the response of the drug? How does the drug respond to the body? What is the, whether if you are taking an anti-inflammatory gel, you have a small inflammation in your superficial area, either in the arm or in the leg, whatever may be, wherever may be, you'll be having a small inflammation. So upon applying the gel, the inflammation might get reduced to a concerned time onwards. So that's what the pharmacological response is of. That can be determined by various processes. Those are acute pharmacological response and therapeutic responses. So these are the two major methods to determine the pharmacodynamic processes. Well, let's discuss about the pharmacokinetic method now. So in the pharmacokinetic method, there are two aspects as we said earlier. The first one is plasma level time studies. So this method is based on the assumption that two dosage form which exhibits superimposable plasma level time profile in a group of subject which results in identical therapeutic activity. So two administration, two different dosage form, when one release is superimposable to the other in a plasma level time profile, so that we can say as a identical therapeutic activity. Mostly in case of bioequivalence, we used to face this concept. With single dose study, so previously we were saying about multiple dose, two different doses. Here in case of single dose study, the method requires collection of serial blood samples for a period of 2 to 3 biological half-life after the drug administration. So their analysis for drug concentration and making a plot of concentration versus time sample of blood collection will be giving us the plasma level time profile. So what is the 2 to 3 biological half-life? So the 2 to 3 biological half-life is nothing but for a new drug when this is an evaluation, or say for example a plasma time study, you are doing it first time for a drug. In such condition, in order to overcome the risk, we have to take the blood sample, collect the blood sample for 2 to 3 biological half-life. In the sense, a drug is having a half-life of 2 hours. Means the drug have the tendency to release in the body for 4 hours. So we have to take the blood sample until 8 hours. Means 2 to 3 biological 2 hours, 2, 4, 6, 8. 8 until 8 hours. What is the reason for collecting until 8 hours? There are some conditions where the drug concentration may be bounded to the plasma. We may not be knowing about it. So the bonding tendency of a drug to the plasma protein might be huge. So in order for 2 hours or 2.5 hours or 3 hours, the drug may be inert in the blood as a protein binded complex. Later, it might get detached and show its concentration in the body. In such conditions, we have to take the drug release from the blood or concentration we have to identify from the blood for 2 to 3 biological half-life for a new drug, for a new dosage form. In order to confirm that the drug as well as the metabolite have been completely got eliminated away from the body. Well, let's move on to the next aspects. So here, in case of IV administration, with IV dose, sampling should start within 5 minutes of drug administration and subsequent sampling should be taken at 15 minutes time interval. In case of oral administration, there is a lag time for absorption so that the blood sampling can be quite different. Whereas in case of IV, the drug release will be immediate soon after administration. So after administration, from the time of 5 minutes, we have to know what is the concentration of drug in the blood and how does it gets gradually decreasing. In case of IV, we'll be doing, if you're giving 100 mg, the 100 mg will be available in the blood. So there is no any absorption parameter, only the elimination parameter. So how come the 100 mg is getting eliminated completely away from the body? Until that, we will be taking the blood sample and evaluating it for the plasma time studies for pharmacokinetic parameters. In case, to describe it properly, the disposition phase for at least 3 sample points should be taken if the drug follows 1 compartment kinetics and 5 to 6 points if it fits to 2 compartment model. So that's what disposition is. Since previously as we said, the elimination process, 2 to 3 samples extra we have to take in case of 1 compartment and 5 to 6 points we have to take in case of 2 compartment models. For oral dose, we have to take a minimum of at least 3 points in the descending and the ascending part of the curve for accurate determination of Ka. So what is the absorption? In case of IV, there was no any absorption process, but in case of oral administration, there was a Ka. So to determine the rate process of absorption, we have to take 2 to 3 points in the ascending phase of the AUC curve. And similarly, the points for disposition or descending phase has to be taken as similar to that of the IV, what we have discussed earlier. Here, there are 3 major parameters that we can able to identify from the plasma level time studies, which is used for determining the bioavailability. The first parameter is Cmax. So the peak plasma concentration that gives an indication whether the drug is sufficiently absorbed systemically to provide the therapeutic response. And the second parameter is Tmax, the peak time that gives an indication of the rate of absorption. So what is that time taken for absorption process that we can able to identify the Tmax. And the third one is AUC, area under curve. So the area under the plasma level time curve, which gives the measure of the extent of absorption or the amount of drug that reaches the systemic circulation. So regarding this graph, a plasma level time profile that we have already discussed in the introduction part of the biopharmaceutics, hope you remember that there will be a minimum effective concentration, maximum effective concentration, AUC, Cmax, Tmax, and we will be identifying about the initial dose or the onset of time and the time phase, which is what is toxic level. All these concepts we have discussed in the introduction topic. Hope you remember. Just to recall the slides which has been given in the slide in the introduction part also. Well, so these three parameters is mainly used for determining the bioavailability that we could come to a conclusion with the help of this plasma level time studies. Well, the extent of bioavailability can be determined by the following equation. F is equal to AUC oral dIV by AUC IV d oral. So this is nothing but the bioavailability as well as the relative bioavailability formula that we have discussed in the previous presentation. So here the D stands for the dose administered, and IV and oral indicates the route of administration, whereas the test and the standard indicates the test and the standard dose of the same drug to determine the relative bioavailability. Well, when there is a multiple administration, so previously we have discussed about the single dose administration. In case of multiple dose study, this method involves the drug administration for at least 5 biological half-life with the dosing interval equal to or greater than the biological half-life. That is, the administration of at least 5 doses to reach the steady state level. So what is the dose required for reaching the steady state level? Until that we will be giving the dose administration. If the steady state is being achieved in the second dose, we will be giving the second dose. If it has been in the fifth dose, we will be going for the fifth dose level. So a blood plasma should be taken, or blood sample should be taken at the end of previous dosing interval and 8 to 10 sample after administration of the next dose. The extent of bioavailability is given as F or is equal to AUC test D standard and T, that is nothing but the dosing interval, test by AUC standard D test into T standard, where AUC values are under the plasma time curve of one dosing interval in a multiple dosing regimen after reaching the steady state, and T is nothing but the dosing interval. So this is a graph which shows the multiple dosing administration, and this is used to determine the AUC and CSS. CSS is nothing but steady state, Cmax, and multiple dosing up to the steady state level. Here there are frequent dosing administration. So in the top of the graph we used to know about dose 1, 2, 3, 4, 5, 6, and for each dose there was a graph which goes as a sawtooth pattern, up and down, peak and valley method. So in the first and the second dose, the dose is been present in the lower level of the steady state minimum. The drug have reached the concentration level. It is within the concentration level. The first dose is also processing the therapeutic activity. It is within the therapeutic range. Second dose is also in the therapeutic range, but the third dose only reaches the steady state concentration level. It start reaching the steady state concentration level, and the fourth and the fifth consecutive doses process a steady state level of drug present within the body. So, and similarly, the area under curve, which has been highlighted with the dashed line. So, area under curve of one dosing interval at a steady state level. So, upon repeated administration, the drug, once it reaches a steady state level, the concentration of the dose will be maintained uniformly. Hope you understood this topic. So, bioavailability can also be determined from the peak plasma concentration at steady state CSS and max according to the following equation. And the equation is FR is equal to CSS comma max to the test followed by D standard and T test by this formula. So this is the formula to calculate the steady state of concentration C max for the multiple dose administration. And the rate of absorption is not important in case of multiple dose administration. So we mostly concentrate on the steady state as well as the Cmax, which is being achieved in the multiple dose administration, whereas we less focus on the rate of absorption. Well, let's move on to the next concept, urinary excretion studies. So far we have discussed about the plasma level time studies, and now we are going to discuss about the urinary excretion study. So this method of assessing bioavailability is based on the principle that the urinary excretion of unchanged drug is directly proportional to the plasma concentration of the drug. Thus, even if a drug is excreted to some extent, at least 10 to 20% in the urine, bioavailability can be easily determined. The study is particularly useful for drug which is extensively excreted unchanged in the urine. For example, certain thiazide diuretics and sulfonamides. And also for the drug that have urine as a site of action. For example, urinary antiseptics such as nitrofurantoin and hexamine. Well, saying about the concentration of metabolite, the concentration of metabolite which has been excreted in urine is never taken into an account in calculation because drug may undergo pre-systemic metabolism at different stages before the drug is being absorbed. So before reaching the systemic circulation of the tissues, there are some chances of drug to undergo metabolites in case of liver pre-systemic metabolism process. So the method involves collection of urine at regular interval for a time span equal to the 7 biological half-life analyzed of unchanged drug in the collected sample and determining the amount of drug excreted in each interval, and cumulative amount of drug excreted will be calculated. So this is what the method preferred for urinary excretion data studies. At each sample collection, total emptying of the bladder is necessary to avoid errors resulting from addition of residual amount to the next urine sample. So that is most important. Complete urinary bladder emptying should be taken, or else the drug concentration in the previous time interval will be getting accumulated to the next time interval. So thereby the drug release means the sampling may be having some errors in it. There are three major parameters examined in urine excretion data for obtained with the single dose studies. Those are dxu by dt max, nothing but the maximum urine excretion rate is obtained from the peak of plot between rate of excretion versus midpoint time of urine collected period. So its value increases as the rate and extent of absorption increases. The second parameter is Tu max. The time for maximum excretion rate is an analog to the Tmax of plasma level time data. Similar to that of the plasma level time data, we used to calculate Tmax. Here similarly Tu max, nothing but maximum excretion rate. Its value this value decreases as the absorption rate increases. When there is increase in absorption, the rate of Tmax of the excretion will be reducing. The last one is about Xu infinity. The cumulative amount of drug excreted in the urine is related to the AUC of the plasma level data, and increases the extent of absorption increases. So this is what the three major parameters which we are going to examine or find out from the single dose study of an urinary excretion studies. So this is a graph which has been plotted for excretion rate versus time, similar to that of the plasma level time study. Here we have a graph of excretion rate versus time. This curve is an analog to a typical plasma level time study, as I said now, and which is obtained on oral administration of a single dose. Here, using this is what we have been discussed in the previous slide. dxu by dt, nothing but similar to that of the Cmax, and Tu max, nothing but similar to that of the Tmax. So the extent of bioavailability can be calculated by the equation similar to that of the plasma level studies. Here we have F as well as F4. We will be calculating the bioavailability using this urinary excretion studies for single dose. Similarly, for multiple dose, the steady state equation has been calculated for using this process. Here, the XUSS is nothing but the amount of drug excreted unchanged during a single dose interval at steady state concentration. Well, bioavailability can also be determined for few drugs by assaying of biological fluids other than plasma and urine. As we discussed in the previous presentations, that excretion of drug through renal and non-renal route. There are chances of drugs to get excreted through non-renal route also. In such condition, we will be estimating the bioavailability through an assay process. In case of theophylline, we use salivary excretion as a parameter, whereas cephalosporin antibiotics appearance of the drug in CSF and bile can be determined. And caution, a major important caution is that the mint, however, being excreted to the account for salivary and enterohepatic cycling of the drug. Some drugs such as mint, which has a process of elimination through two different routes. So by estimating either one parameters, we can't say that only 50% of drug got eliminated. So we have to consider the other route of elimination also. Well, let's move on to the next parameter of this studies is about pharmacodynamic. So far we have discussed about pharmacokinetic. Under pharmacokinetic we have discussed about urinary excretion as well as plasma level time studies. And in case of pharmacodynamic method, the first is about acute pharmacological response. When a bioavailability measurement by pharmacokinetic method is difficult, inaccurate or non-reproducible, this acute pharmacological effect such as ECG, EEG, pupil diameters, these can be taken into account. So this will be taken into account and to the time course of the given drug. So if you are administered a drug and we are expecting a response in 15 minutes, so we have to identify within 15 minutes does the response has been found or not. In case of acute pharmacological response, we can't go with the major pharmacological response such as hypertension or diabetes. For all these things we need a estimation of blood or some tissue studies. We need all these things. These are few aspects which we can know by simple experimental studies, ECG or EEG or pupil diameter such as mydriasis, MEOS, all this process. This method requires measurement of responses for at least 3 biological half-life of the drug in order to obtain a good estimation of the AUC. To confirm the AUC better way, we should take at least 3 biological half-life of the drug. There are some disadvantages in this method. Those are there are more pharmacological variable responses chances during the study. During the study, there are some chances for more variable pharmacological response. Sometimes there will be a immediate response. Sometimes there will be the response will be later or delayed due to several physiological conditions. And the second thing is accurate correction between the measured response and drug available from the formulation is quite difficult. We can't exactly do the correction, or we can't exactly identify the differences. In first dose, it might show some response, and the second dose, the response might be quite different. As I said earlier, might be due to some physiological disturbances or variations. The next one is about therapeutic response method. So this method is mainly based on observing the clinical response to a drug formulation given to the patient suffering from disease for which it is intended to be used. For diabetic, in the sense, what is the response of the drug for diabetes? For hypertension, in the sense, what is the response of the drug towards hypertension condition? And that is what about therapeutic response method. Even this method also have few disadvantages. One is quantitation of observed response is too improper. We can't able to quantify it. There will be some mismatching or not correlating to the free every sample in case of quantitative method. And unless multiple dose protocols are employed, a patient who requires the drug for a disease would be able to receive only a single dose of the drug every few days or perhaps each week. So this is the second one. Multiple dose protocol will be used to maintain to get the desired concentration. So this is the other disadvantages of the method. The last one is about many patients receive more than one drug, and that results obtained from the bioavailability studies could be compromised because of the drug-drug interaction. So without the knowledge of the physician or the pharmacist, they might take two different medication which have interactions to each other. So they might get one medicine quite long earlier for some response, some diseases, and currently they might be going for some medication process. During this medication schedule, they might have a symptoms of fever or headache which they have been earlierly encountered. So remembering or recalling the earlier medication, they might have that in their home, so they might take the medication now along with the existing diseased condition or existing prescribed medication. So there might be chances of interaction of these both the drug which may cause a therapeutic ineffectiveness or else sometime might lead to toxicity also. So this has to be taken under consideration. So this is what about the pharmacodynamic method. So in today's class we have discussed about two major parameters used for measuring the bioavailability. One is pharmacokinetic. Under pharmacokinetic we have discussed about plasma level time studies as well as urinary excretion studies. And the second we have discussed about pharmacodynamic method. Under this, acute therapeutic response as well as therapeutic response method we have been discussed. Acute pharmacological response. Others, second one is therapeutic response method. Hope you got understood about the concept in detail. In case of any clarification, please don't hesitate to contact me back. Well, To conclude, let me use this quote: life will never provide warranties and guarantees. It can only provide possibilities and opportunities to convert them into success. Life is not an electronic device, right? So for electronic device, they used to say warranties and guarantees. Life is not an electronic device. We can't have any warranties or guarantees for what all the things we executed of. Well, in case of the current scenario, this corona effect, no one predicted that on Jan 1st, this year will be like this, or we will be getting locked down, being stagnant in the home for 3 months, whatever may be. We have not predicted, no one. We started the new year with a new hope. But unfortunately, there was a critical condition. But even in the critical condition, we are overcoming with our abilities. We are overcoming with various aspects. In case of education, we are using an online platform. It's a new one. So recently there was a talk I was in attend. In the talk they said that every natural calamities or every task gives a new resolution, a new revolution. So we are focusing that and we are overcoming the problems which are facing now. So this is one thing about it. Well, coming back to this topic, there is no any warranties or guarantees in life. Only we have possibilities and opportunities. There is opportunities in every possibilities, and there are possibilities in every opportunities. Whatever you're facing from now, think that properly and find an opportunity in the event what you're facing of. Similarly, in the opportunity what you're getting, grab the opportunity and find the possibilities to convert it into a success. With this, let me conclude the today's session. Thank you all. Thank you all for your patient listening.